Welcome back to CEC Gurukul lectures on bioinformatics. We've been talking about how to build a phylogenetic tree from the sequence data which we have, and we talked about the several concepts in phylogenetic analysis, and in this section we just learn who is who, and in the phylogenetic tree how do we draw in this section after the break. So, before we go further, a quick recap what we learnt earlier. In the phylogeny study, we try to build a tree in which we study the relationship and origin evolution of the species. The tree can be rooted or an unrooted tree, and distances among the species are the key for construction of phylogenetic trees. We have distance based trees, we have character based trees, and the UPGMA method and neighborhood method is the nearest neighbor method is the distance based methods, and we have maximum parsimony and maximum likelihood are the character based methods. We have popular softwares like Phylip and Mega and RAXML with which we are going to build a phylogenetic tree. So, let us start with building a phylogenetic tree. And building a phylogenetic tree, we have few steps involved in building a phylogenetic tree, and after talking about the steps, we will talk about our sequences with which we are working. Then I will tell you what all softwares are available for making a phylogenetic tree. We will explain the result of our sequences, that when we build a tree with them, what result did we get. And we will also talk about how to decide which is the best method for building the phylogenetic tree. So, first talk about the steps involved in building a phylogenetic tree. So, the steps involved, first step is we choose the appropriate sequences for building a tree. So, meaning there should be some difference between them, as we have been talking earlier in the previous MSA class also, there should be some difference in them. We will make the multiple sequence alignment with these sequences, then we select an evolutionary model and we will make the phylogenetic tree, and then we will evaluate this tree, that what is this tree showing us. So, first the next thing is we wanted to talk is about the sequences. Like the sequences which we selected for today's work are the 10 sequences. They are viral sequences, they are named as GP120, and they can be divided into two groups based on their transmission. Like GP120 sequences that are crucial, these sequences are crucial for binding to the host cells. Host, this is host, not ghost, right? And what happens is the only partial length is selected, and we have two series. This is what I was trying to focus here. In these sequences, we have two series, one HIV1 that arises from cross transmission from chimpanzee, and we have HIV2 that originates by cross transmission from the Mac monkey, right? And on the right side it shows you a notepad with which where we have saved all these sequences. So, the two sequence sets are expected to belong to two different clades when I am going to make a phylogenetic tree, because they have come as a result of transmission from two different organisms. So, we will first do the most easy method. We try to make it do the multiple sequence alignment with cluster omega, as we talked in the last class. We will accept all the default characters, and then we get an alignment file. You can see the result summary tab there, and then you see the alignment file, and then you get the tree file, and you also get a similarity matrix. In the output you get everything, and this is the alignment. If you click the alignment file, you can see the alignment, and this is the tree file. And the point to be noted here is we did get a tree by cluster omega also, but it did not give us two clear clades. If you see it clearly, it is more than two clades. So, this tree formation by cluster omega was not basically on the algorithms for making the phylogenetic tree. So, it made a tree on the basis of its program, but our sequences did not give us the right clades. So we took those sequences, and I told you we have Treeview software, that is treeviewX. You can download that on your laptops or machines, and it's a standalone. It works very well. You can observe different meaning your sequence, like the tree it had drawn in the cluster. You can observe this in slanting form or maybe rectangular or whatever way you like. It gives you several options to view a tree the way you like it. So that was about cluster, building a tree, the simplest method, and viewing that in the tree viewing software treeviewX. Now we come to the next program, that is Phylip. Phylip is also a very popular phylogeny building software, and it is derived, Phylip is phylogeny interface package. This was developed by Joseph Celestine, and it is written in the C language. It is freely open access, it is freely available, developed from University of Washington, and it is basically, it's a big software, and it is basically a package of 35 portable softwares, and they are implementing different meaning different methods, like we have parsimony, distance matrix, likelihood. All methods are being used for building the trees. So we have 35 different softwares in this package, and you can choose whichever way you want to make it. And you also have bootstrapping and consensus tree building softwares in this package. So the data types that can be handled include molecular sequences, gene frequencies, restriction sites and fragments, distance matrices, and the discrete characters software. So you can it can use different forms of data, and like for protein sequences, if we talk, it provides us several softwares in the package. So we are not using it here for our sequences, but I had tried it with some more sequences, meaning instead of 10 we run this for 20 sequences, but it clustered very well. It clustered very well. Because of technical difficulties, I could not show you all steps here, but I will show you the steps for MAGA, the detailed steps for MAGA, and compare the tree drawn by MAGA to that of clustral omega and that we viewed in Treeview software. So MAGA is the stable version of this, has come in 2018 recently, and it was developed by originally, but now the stable version has come in Pennsylvania State University, and it is quite popular and quite easy to use. Like I show you, that's a small video of my laptop, where I was just trying to show you the MAGA software, how it looks like. So I have downloaded that on my laptop, and you can select and to build a tree, and you say you are building a new tree, you open your sequences from your laptop, maybe it is opening it from my laptop, and then it will open now sequences from my laptop. It will ask me to locate the sequences, and now it takes up the sequences. I have stored these 10 virus sequences there. So it opened up those. Then I'll ask you to select all and make a alignment, and it would ask you whether you want alignment on the basis of cluster or you want alignment on the basis of muscle. So I selected cluster, so it made an alignment for me on the basis of cluster. Now I would ask it to make a phylogenetic tree for me, and it made a phylogenetic tree. It took the data for building a phylogenetic tree, and now I would decide which method I want to use for making a phylogenetic tree. So I can make it by ML method or NJ method. It provides me option for all the methods. So in this small video, I was just trying to show you that I can make trees. So what I did is, in quick steps, I made it for all 4. And after this is doing for all 4, I have saved those figures, and we can have a look at that, what type of trees have been drawn by the 4 methods. This MAGA software also allows you to select data online, meaning it will allow you to go to meaning search by the internet sources, then you can go to NCBI, or maybe you can go to the other protein sequences, and then you can directly select the sequences and run your data. So, here is a tree which is drawn by maximum likelihood method, and you can see that we could make 2 clades. Our data belong to 2 clades and it made a clear 2 clades. But what happened with this is MP method, and we do not see 2 clades here, and in this one also we could get a UPGMA method, we could get 2 clades, and it just did fine. But it was very easy. The point to be focused here, it was so easy, and you see the evolutionary distance written, the similarity in the key to at the bottom of this diagram, and you could very easily make it for the sequences, and there were differences. And did you observe the tree made by Clustal? Also we couldn't get it in 2 clades, but here we could get it. But because these were very small sequences, meaning on only 10 sequences we were running, we could do it like a fun. There were no problems arising. But problems came when I was trying to run make a phylogenetic tree for my sequences, which were 237, and they were very long sequences. So, I needed lot of computer time and lot of accuracy in these ones. So, what we did is, rather than using this mega, I shifted to RAXML, that is randomized accelerated maximum likelihood tree building program. It was available online at Cyprus, because they are supercomputers. They allowed me to submit my data and select the parameters which I want to use. Like I said, I want to use 1000 bootstrap, and I selected ML method, and I also had few more choices in deciding that how I wanted to run my phylogeny. And what it did is, it did it and it sent me the tree file, and for viewing the tree file I selected another software which allows me to build huge phylogenetic trees, like it is Ital. And I tell you, this Ital not only allows you to view good trees, make good printable forms, like when you are going to submit it for a paper or anywhere, it also gives you several options, meaning you could change them, you could delete any row whichever you want is creating problem in your tree for viewing, and you can view any form of tree with this one. So, this was about Ital, and I told you 2 softwares for tree viewing, that is the website, and Ital is for making super cool trees. You can make very beautiful trees with Ital, and that was about my data. And when you reach to the larger data, you realize the importance of having faster methods and having the accurate methods at the same time. So, that was about building a phylogenetic tree, and I hope you could appreciate how important it was to understand multiple sequence alignment before studying the phylogenetic trees. So, I will just quickly summarize before I end, and I tell you the references from where we can study. You can study this in details. Is that we talked about phylogeny, where we talk about relationship between the species, or maybe we try to build a tree which explains from where it originated, meaning where is the root and what are the branches, which route it took for evolution, and where are the leaves, meaning present day species. We have different types of trees, rooted and unrooted. I showed you those with very good examples, and it was quite easy to understand the concept of outgroup. And with the help of my sequences, I showed you how important it is to decide a proper outgroup, because it has to be from an unrelated sequence belonging to unrelated taxa. We have to have differences among species if we want to build a construction phylogenetic tree, because the difference is the key for building the tree. We have 2 types of methods for building phylogenetic trees. They may be distance based methods or they may be character based methods, and we studied all 4 methods with the help of Mega software. And the popular softwares, I said Phylip and Maga, and Maga was very easy to use. I run one program with few sequences of hours, and it really worked well with Maga, and you don't hardly need any training to train yourself with Maga. You can download that on your laptop and work with this. We have another popular, like the other popular softwares is RAXML, which I really found handy to work with. It gave me quite accurate results, and it could give me several options to decide, and it handled huge amount of data of mine to run a phylogenetic tree. We have several tree viewing softwares like TreeviewX. I told you it is very simple, very easy to use. You can download, it's freely available. You could download this on your PC or laptop, and you can enjoy different views of your sequences. It is very simple to work with. But if you are going to build super cool, colored, beautiful trees, you can use itol for this purpose. Itol provides you a lot of flexibility, meaning you can change colors of the branches, leaves, you can provide them a label, and you can just move one leaf from the other to see the relationship, meaning if in theory if you know that this branch should come here, it will pick up your branch and you can just change the branch location. So you can fiddle with it a lot in itol and get result of your choice. But it was really good, and it's an online software. You don't need to download this, and you can work very well with this. So we also learned that the ML methods are quite accurate, but they are very time consuming. They require a lot of software computer time, but they were accurate for working. So with this one we have finished talking about, but before we disperse, I would like to tell you some good important references where you can find the information about this phylogenetic tree building in a very easy and simple way. Like I told you in the last class, the book of Mount, that is Bioinformatics Sequence and Genome Analysis, that is one of the Bibles for studying this part. So if you follow Mount book, Bioinformatics Sequence and Genome Analysis, that is really good. Then I have added two links here, that is I have given the path, and maybe in the description section of this video I would add these again so that you can follow these two paths. They are very good write-ups by two great scientists who explains this phylogenetic trees building process in a very simplified and a clear manner. Then you can also look at this book, Basic Informatics. Another reference which I thought I must include here is Introduction to Bioinformatics, a book by Atwood that describes, that is especially good for phylogeny building. It gives you the meaning, especially if you are a mathematics, if you want to look at the statistics of this, I would advise you to have a look in this book, that is Introduction to Bioinformatics. So with this one we come to an end of this talk about phylogenetic trees, and I hope it was quite interesting and you could understand whatever we've been talking till now. If you have any questions, we'll be very happy to answer your questions. Thank you very much. Thank you for your patient listening. Thank you.